Okay, so today we are going to talk about module 5, that is proteins. And we are going to learn about, first of all, how they are derived from DNA. So studying the process of transcription and translation briefly. And then we will talk about the different levels of structures of protein, that is primary, secondary, tertiary, and quaternary. Then this will be followed by the characteristics of different enzyme categories and what are their functions in human. So as you know that last week and even before that, we have been talking about how the DNA is converted to RNA. And in specific, we talked about its conversion to messenger RNA. So this process of conversion, I just want you to see this figure. And if you see here, the DNA, which is double-stranded, it has to be unzipped or opened up. And then it is not the 5' to 3', but the bottom or the in this figure, or the 3' to 5' strand, which is actually used to carry out the process of transcription. So essentially, once the messenger RNA is formed, there are other things that have to happen, which we are not discussing in detail because it's not a cell and molecular biology style of course. But just for your information, sometimes the messenger RNA is directly translated into proteins, as that happens in many bacterial or other microorganisms or different yeast systems. All of those, there can be a direct conversion of messenger RNA to proteins. However, it is also possible that sometimes, or more or less almost in eukaryotes all the time, that messenger RNA may have some extra coded sequence. So they have to be processed, and then that processed messenger RNA goes in the cytoplasm where it is converted to proteins with the help of ribosomes. And as you probably are aware, ribosomes can be also present on the endoplasmic reticulum. That's why we call it ARIA, or a rough endoplasmic reticulum, which contains the ribosomes. So this is the typical structure of a ribosome. If you see this in this yellow orange kind of structure, that's what the ribosomes are, and shown hereby. And then there's a messenger RNA. And you can see that this figure doesn't do complete justice to how the RNA is converted to protein. But if you see here that the chain, the green chain, which is the representative of the protein chain here, keeps growing, and then eventually it gets released. And as it gets released, it is actually, that is the protein that is eventually folded by the cells. So if you see on the left-hand side, it just briefly states the process. DNA is converted to RNA by a process called transcription. Inside the DNA, and then the RNA is converted to protein by translation. Okay, so then let's see what happens after that. Okay, so after that comes that process where we were talking about it, and that's where I'm going to allow you to, definitely ask questions which are relevant to module 4. Of course, you can ask anytime. And so in this one, if you see here, the DNA is converted to RNA using base complementarity rules. So we have to know these rules. These are also called Sharkov's rule. And, this Sharkov's rule is something that we have learned, that is A pairs with U, or the complementary base, not really pairs, but the complementary base for A is U, and for G is C. So you can imagine that if it is, A on DNA, then it becomes U on RNA, as shown by the very first base pair here. And then the next one, if you see here in blue, and I'm talking about the figure on the right-hand side, bottom panel in the cream color, right? So where my mouse is. So the C is converted, or C is transcribed as G. Similarly, C here becomes G, A becomes U, and it doesn't show everything. So on the left-hand side, if you see T if there is a T, it becomes A, and C becomes G, G becomes C. So there is this linear correspondence which converts the DNA to RNA, or messenger RNA, as we say, and that is the process of transcription. I want to pause here before we go to translation and ask if there are any questions which are related to transcription or the conversion of DNA into messenger RNA.
So after transcription is done, when we go to translation, do we just group every three? We just check every three letters and see which one they join?
That's right. And your question is very good, actually. Your question is good because that's what we are doing here, and that's what we do when we learn this. But if you say what happens in the human body, that does happen, but the process of translation, there are other proteins that are involved that will come and help. It's not an automatic process. So just that clarification, none of these processes are happening on their own. They are happening with the help of a bunch of proteins which come together. And they are responsible for carrying out the processes. So for manually doing it, as someone who has to convert the RNA to protein, yes, your answer is absolutely correct. You are bunching them in group of threes. Now you can also say, you grouped UGG. Why didn't you group, like leave U and the start with GGU? And that is a very valid question. But the kind of questions that you're being given are just for your basic understanding of transcription and translation, which means that you will be actually just clubbing three times each time. And what you call those three is a codon. And that codon is the one that will give you the right amino acid. And I really, really want you to understand this. I know that you may not be doing a whole lot of molecular biology stuff. This is extremely important. I'll give you a very small example. Have you heard of all the virus mutations that they keep talking about, variants of the virus in the news? So if,
Yes, we're related to COVID.
yeah, yes, you're right.
They have like the new one, like the Brazilian and the European virus.
Yes, all those variants, that's how they come, because there is sometimes a single mutation based variation in DNA. So if you see here, so I'm going to mute you guys, but you can unmute yourself anytime, So what happens is, if there is one mutation, let us imagine for once that instead of C pairing with G, let us imagine for once that it has I hope my computer doesn't like give away, because it's like in a crazy state today. Okay. So let us imagine for some reason, and you may say, how can that even happen? That does happen. Okay. So we are going to imagine that there is a T here. I'm sorry, not T, I'm sorry. I meant U. Oops. So imagine that there is a U here. Okay. So if instead of G, accidentally a U was put here, can you tell me UGG is coding for a tryptophan, but what will UUG code for?
Leucine.
Leucine, very good. So that means that we are going to be not having the tryptophan anymore, and that will be replaced by a leucine. That makes a very different protein. The properties of tryptophan are different, and the properties of leucine are different. Tryptophan may be interacting with something which leucine may not be able to, or it can interact with something else, giving rise to a different protein structure. That is why when there is one mutation, transcription, or while there is in the process of DNA duplication or transcription, when there is one thing that goes wrong, it can give rise to a protein that looks actually different from original protein. And then that protein may be responsible. And imagine that protein is on the spike protein of a virus. It's one of the spike proteins. So what happens is, it results in the virus looking different. And that's what we are calling a variant of the virus. So these codons are extremely important. And these variation can result in protein structures which are going to be completely different. And actually that's what I'm just relating it to your bonus assignment. I'm going to put a bonus assignment which you can feel free to complete or not complete. It doesn't take away any points for you, but it's...
Wait, is that available already?
No, it's not there. So it's going to be on sickle cell anemia and how, that one protein change will lead to a change in the shape of hemoglobin, as a result of which the RBCs, instead of looking round or more like what should I say, a disc shape, they're going to start looking like a sickle shape or more like a C shape, right? So
Oh yeah, for anemia, I think like they look like usually like the red blood cells look like a circle, but like when people have anemia, they're like halfway, kind of like a crescent moon.
yeah, absolutely. I think that's Valerie speaking. Okay, so yes, you are absolutely right. That happens. It's a single mutation, a single base mutation, which leads to that. That is why it is so important to learn. It is directly related to your fields. You're going to be in either nursing or dental hygiene, and you will be learning about some of these health conditions. So you should know that single mutations can lead to certain health conditions. Now, focusing on the translation part, I just want to know that I don't want to repeat myself because we have done this two weeks consecutive. Do you think you can, in a codon, can you convert it to the amino acid which you can put in place of, just once you translate, can you put it in the sequence? Do you think you can do that if I give you a certain codon, let us say you should be able to locate that as U, and then point it out as phenylalanine and just put it here as PHE. You can put the whole thing or F, whatever is on the table given to you during the exam. But do you think you can do that comfortably?
Yeah.
Yeah.
Okay, and some people said that during the exam, they were not able to upload the question number, some question related to this. And I said, yes, you can send it, but I'm going to go back and try to see if I can fix it so that others who are doing the exam will not have this problem. But of course, if there is a problem during the exam with about something, you can message me then and there. And sometimes I try to respond right away, but if I can't, then, I mean, it's not your fault. You won't be penalized for something that, it's not your fault. So you please don't be thinking that if you couldn't do a question just because you couldn't upload something, that I would take away your points for that. Okay, so with this, I want to go to the next part. I hope this was clear because there is only so much time we have, right? So here is just a pictorial representation. Again, it's not in a whole lot of detail, but just because it's an introductory biochemistry class. So there is this DNA, which is 3.1 billion base pairs in every nucleus of every human cell. And out of that, there are certain parts that are coding for a gene. So we discussed only 1 to 2%. And that gene is shown here. The gene there is a messenger RNA that is generated. Once the messenger RNA is generated, actually they should have just shown it in a different color. But once it's generated, it is actually going out of the nucleus into the cytoplasm, where it is converted to a protein. How does that happen? It is it is attached to the ribosome, and then these ribosomes, they will be different in prokaryotes and eukaryotes. As you know, we are eukaryotic organisms, right? So there is in the ribosomes, what happens is that it's not able to do it by itself, but there is additional help. So ribosomes are made up of protein and ribosomal RNA. Once there is messenger RNA, pairing with the messenger RNA, there is these codons, and who is bringing those amino acids, which are, let us say in this one, you knew that there is a phenylalanine, or the first one is tryptophan. So who is going to bring that? That's going to be brought by a transfer RNA. So you see this odd shaped, like a clover leaf kind of structure, and it will be carrying these green parts are the free amino acids. Transfer RNA will bring them, And once they are brought, they are also connected by a peptide bond. And then the chain grows. And once it reaches the stop, it's going to release the peptide or the protein chain. And when I say peptide, I mean a smaller protein. I don't really, I mean, it's essentially the same thing, whether I say peptide or protein. So this results in the process of making of the entire protein. And so that is the goal. That is the goal in the sense, like whenever there is external stress or there is growth happening or anything that needs production of protein, it has to follow this process. Of course, there's a lot of processes that can influence transcription and translation, which you might study in another class. Okay, just so now onwards, we can hopefully go a tad bit faster. Proteins are polymers of amino acids. Why are they called heteropolymers? Because hetero is different types. And if you see here, these are the amino acids that are connected. They are joined together. So amino acids are the structural units that they're the monomers. So just like there were nucleotides, there was monosaccharides, and there is amino acids are the structural units here. They can be represented in different ways. You know, we always try to study them by looking at different kinds of ways of representation. And the reason for that is that you might encounter any across while doing some other class or while you are in your field, right? So there could be chemical formula or structural formula. We are going to probably stick to this format the most. And if you see here, you can have a ball and stick model. So please, if you see these, they mean exactly the same thing. So all the amino acids, they are, essentially they are chiral except for glycine, which is the first amino acid that you would study, and you will see why, because the R is H. So they have essentially 4 different groups. So you can, if you remember the concept of chirality, you really don't have to create mirror images. As long as you know that there are 4 different groups, you'll be fine. There is a negative charge due to carboxylic group, and there is the amine group creates a positive charge. And once you have the negative and positive charge together, that's called a zwitterite. That's what is also written on the left side. Also, all naturally occurring amino acids are L amino acids, and all the sugars are always D. And we will talk a little more about this. Yeah, just to just saying something funny. Okay, let's get to this. So amino acids are referred to as alpha amino acids because because of the nomenclature, that carbon that it is attached to, it's called the alpha carbon. And they can be of different types. So instead of going through each of these definitions, let us actually see the structures. We do know that nonpolar is hydrophobic, or it doesn't like, it does not like water. Polar is the hydrophilic, or it is has affinity for water. And then there is uncharged, and then there is charged. So these are the different just ways of, grouping them. Okay. So let's focus for 1 second on the left side. We are going to be talking about non-polar side chains, polar side chains, and electrically charged side chains. So if you were doing a more intense biochem course, it was a 200 level course, then we probably be looking a little more details. But you don't have to memorize this, even though with this it might become a tad bit easier. This just way of saying it, and then you can divide the amino acids in different groups. But you don't have to do that. It's just that a way if you want to do it, but you don't have to do it this time. You won't be asked in the exam suddenly like, okay, which one of this is, like name all the non-polar side like amino acids, and that won't happen. Okay, so non-polar. How do we identify amino acid as non-polar? Extremely important. I hope you are focused, paying attention to this part. And that would be that whenever you see it's long chain, it's carbon, not even long chain, like if it's carbons only, that's non-polar. Carbon doesn't really, until there is a functional group attached like that, carbon really doesn't really like to mix with water. So if it is like a, long chain fatty acids definitely don't. So any kind of carbon structures are always non-polar. That's what it is here. And of course, this is not something that you've done before. So just clarifying, this is a benzene ring. And if you see here, that's also two fused ring structures, and these are all non-polar. Now, let us look in contrast why these are polar. These are polar because they have, in addition to the carbon, they also have a hydroxyl group or a sulfhydryl group. So this is oxygen with hydrogen, this is sulfur with hydrogen. So that's what it is. And that's a combination of nitrogen and oxygen. So any of these would make these polar. Essentially, what's going to happen is once you reach the assignment, you're going to be asked questions about R or the side chain. And that R or the side chain is, is this one, the one in the green or the one in the purple? That is what the side chain is. And what is the functional groups are right here, okay? Okay. So what about the electrically charged side chain? So you can see that there are some more variations in here, or there is acidic and basic group. Why do you call this acidic? You can see that this already had a COO as a part of the amino acid, but there's additional COO group in two of these, that is aspartate and glutamate. You will also see the name aspartic acid and glutamic acid. And similarly, when you are looking on the right-hand side with these blue ones, you can see that additional NH2 groups, which of course in a ionic form would show plus NH3, and that would be your extra amine group. So that's more basic, having more of this style, and this is more acidic. So, I mean, in the sense, so you can see how important these things become in structure. Suppose there is something coding for aspartate, and that is negatively charged, and due to any mutation, if it starts coding for like a arginine or asparagine, or for that matter any other one, then they will no longer have a negative charge, and the protein will start looking very different. So you have to really appreciate how the nature has really coordinated this whole thing, how few mutations are there. There can be a lot of mutation. There are 10,000 mutations per cell per day, and they are extensive. DNA repair fixes it, but I'm just saying there's a lot of possibility of errors that can happen. Anyway, coming back to this, does anyone have any questions about non-polar side chains, polar side chains, or acidic or basic side chains within the polar side chains, of course?
I got a question about like the electricity charged ones. So down, you're saying that if it has like on the first one where your mouse is, where it has like the negative O, and then if you look on like the other side where you have like the positive, like the NH3, that's how you tell if it's positive or negative.
Yeah, that's right. And we also know that this is a basic group. We already know that if you see a carboxyl group, that is an acidic group, and a amine group is a basic group. We know that it has a tendency to get one more H and have a positive charge, and this has a tendency to get rid of that, especially in, these are not alone. They're going to be in water, so they're going to be charged because of other factors. But you're absolutely right that whenever you see two of NH3s, that does indicate that that is a more basic. And whenever you see more of COO, that is a more of carboxyl. So great question. You can see that how in histidine it can be a little bit deceptive. You can be like, wow, that is, that structure looks very different. Also, you might question me and say, hey, I see an NH2 here and an NH2 there. Is that not more basic? And my answer to that is yes, that is, I mean, that can be, but if you see here, there is a C double bond O as well. So the charges are not really very, what should I say, like not always well defined. It also depends on where the amino acid is. I know we're talking about cytoplasm, but depends on what charges it is subjected to. So the positive negative charge itself is part of the polarity. So these slides are just like the repetition of the same thing. that was so small. So there's just bigger for you to be able to see. And when you're doing assignments, it's kind of helpful that you see that. And in assignment, there may be questions like, if you see this versus this side chain, what is the difference? So just, go back and take a look if you need to. So I'm skipping this one. These are the 3 different ways of representing amino acids. So I think we had a question in module 4 because we were talking about more about DNA and not talking about amino acids at that time so much. So I did not share the table in that module, but you can see here that there is, it's not all obvious. Example, isoleucine, well, that's not iso. And similarly, when you're talking about, tryptophan, that is not TRY, it is TRP. So it's pretty close, but not all the time. Okay, so there are essential and non-essential amino acid. I personally think it's a useless exercise to try to memorize all of this because, learning is changed a way lot than before. But what is to be remembered is that essential amino acid must be obtained from the diet. So they cannot be, they cannot be synthesized by human body. So if you see here, methionine, threonine, valine, phenylalanine, isoleucine, tryptophan, lysine, leucine, all of these. And so, yeah, I think this is a pretty neat way and very easy. My tall vegan friend is watering kale leaves. It's a nice way to remember, but you have to remember all the amino acids. So like if I was to memorize this, which is not that difficult, I would also need to know this, which I know, but I don't expect you to memorize all the 20 amino acids. So all I'm saying is there is a way to do it. More than that, what's more important is essential and non-essential. And you can see here, there is more classification just for your information, not that we are going to use too much of that. So one of the most important thing that we are always focusing on when we are studying macromolecules is how are they combined together. So extremely important for you to know, again, this synthesis, it's a condensation reaction, which means that the water will be going out and there will be a combination or there will be a bond that will be forming between CO and NH, and that CO-NH bond is called the peptide bond. So if you see here, this is something, if you remember, like when we were talking about glycosidic bond, we said the exact same thing. When we were talking about, how glycerol and fatty acids come together, again, the same thing. So it's a very standard process in condensation and hydrolysis by breaking with water. And again, condensation means that water is coming out. So if you see here, there is also 2 labelings here. N terminus is the start, which you can see here. This is because you're facing an NH3. And ending is the CO minus, so that is the C terminus. So just getting you used to the nomenclature. This is the peptide bond. It's between every amino acid. So if you see here, there is this blocks of gray, and these are the CO-NH bonds, C-O-N-H, C-O-N-H, that's what it is. And if you see here, what is most important is the R. This R will be varying based on the amino acid, it varies. Leucine is different, tryptophan is different, I don't know, aspartic acid is different, valine is different, all of these are different. So this is how it works. When you put it together in a very simple language, you can see that this is how it's going to look like, okay? So if you see here, there is a dipeptide, of course, you can understand that di means 2. And so these are the 2 residues coming together, it forms a dipeptide. 3 residues coming together is a tripeptide. And then several of them coming together, not really like 100, but like more like 12, 15. Those are oligopeptide. Polypeptides are 50 residues and proteins are more than 50 residues. So essentially, these are just different ways of representing the smaller peptides, I guess, the smaller polypeptides and oligopeptides, all of these. Protein structure is characterized at 4 levels. There is primary, secondary, tertiary, and quaternary structure. So if you remember when it was coming out, it kind of comes out like that, but then it starts folding very rapidly into secondary and tertiary structures. So what is a primary structure? It's just simply amino acids joined together. So what I have been showing you so far is the primary structure because it's kind of easy to understand and you can see that it is just connected together. And you can see or also appreciate here there is an N terminus end and then there is C terminus is not shown here, but it would be in the other end. I mean, there is an N and C everywhere. This like, starts with N, C, N, C, N, C, like that. And so that is why it ends on C. Okay, so now there is secondary structure. So important to recognize that these chains will sometimes, a lot of times actually, would have a repeating pattern, which means that they will form hydrogen bond. And I think that hydrogen bond should remind you of what you read in module 4 and how the bases will come together. So the hydrogen bond can result in, and this kind of bonding can result in two different types of structure primarily, which is one of them is alpha helix and the other is beta sheets. If you see here, the alpha helix and beta sheets, even though both of them have hydrogen bonds, they are a little bit different in the sense that, you know, you can see that this is more like parallel structures where it is occurring at a certain interval, and similarly here, same thing, but it is more like looking like a helix. And so these are two possibilities. So what happens is, if you see here, I'm trying to see if I can show you here. I see more of alpha helix in here. So, I mean, I'm just trying to see if I can get some similarity, but these alpha helix and beta sheets, they'll come together in a more three-dimensional kind of form, and they will be forming this three-dimensional form, not only just because of the hydrogen bond, but there can be additional things. For example, there can be a disulfide bond. And so that disulfide bond is the one that brings two S and S together, and that is the two cysteines together. So all I'm trying to say here is, as you go to the levels of protein structure, it is important to realize that these proteins may have not just the peptide bond, but a little more than that for them to come together and have a certain shape. And what happens when it doesn't have a certain shape? It cannot do what it is supposed to do. So it needs to have a certain shape. Now imagine there are 30,000 proteins which result from your genome. And so in every cell, how many ever proteins are generated? Not every cell has all 30,000 being generated. But what I'm trying to say is, it's really important to recognize how body can make sure, or how every cell can make sure they're folded properly so that they're doing the right functions in the body. And if that doesn't happen, that is always associated with then diseases or conditions which are, you know, which are important to recognize. So this is important. And then there is quaternary structure. So the protein consists of more than one amino acid chain. So you can see here, what does that mean is that this is one big chain, and there can be 4 different chains coming together. And each of this chain is shown in different color, just for you to be able to recognize the 4 different chains. And actually that would be like the structure of hemoglobin. So there is not just one chain making one protein, sometimes multiple can also. There is no hard and fast rule. There can be trimeric, there can be tetrameric, there can be dimers, which means dimer means 2 chains coming together, tri means 3, and 4 means tetra means 4. So there can be all these different variations. All you need to know is essentially that there are 4 different levels, and these 4 levels are resulting in a protein structure and shape that is active, and that will result in a certain function happening on a certain structure happening. And we're going to very quickly see now the roles of these. So if you see here, we were just talking about this. There are multi-chain proteins. They are held together by the same forces as the tertiary structure. That is, there can be hydrophobic forces, there can be hydrophilic, There can be positive or negative charges. There can be cysteine interactions. All of these keep them together. And if you see here, there is this potassium channel. This is 4 identical chains. That's an antibody. You have heard about antibodies a lot because, you know, antigen, whenever there is antigen, the body will respond by generating an antibody. And then there is another G protein, which is 3 identical, non-identical amino acid chain, just showing you the variations. I hope you can appreciate them. So when you are looking at structure of a hemoglobin, you can see here that this is what is present in the red blood cells, and is responsible for the transportation of oxygen. And what hemoglobin does is a dual function. It, you know, it supplies the oxygen. It also takes away the CO2. So whenever they talk about making artificial blood and the advances they have in making artificial blood, the challenge they have is they always can make a molecule that can deliver oxygen, but at the same time, that particular protein is, or whatever they're making, it's not able to take away the CO2. So just reflect the complexity of biological molecules and the functions that they can perform. So for you to see that it's not just simply a chain, which is how it is represented, but a more complex structure which can carry out different kinds of function. So why are proteins important in the body? And this brings us to the part where it gets a little more like easier to understand. So I'm again going to go a little bit faster because I do want for us to finish on time. So feel free to interrupt me if you need to. So proteins, they can, you know, they can do a lot of different things. For example, they can be used for structural part. They can be used as, they form majority of the enzymes. They defend the body as, because their antibodies are actually nothing but proteins. So they defend against the foreign elements, and they can just perform so many different tasks. They will also serve as receptors on the membranes, and they will help a certain signal that is given to the cell to be amplified inside the cell. So just a lot of different functions, and we are going to just study the basic functions here. The proteins can be globular or they can be fibrous. What do we mean by that? Because this is the first time you see the term globular, but it's kind of obvious that they have a globe-like or a round shape, and these are typically water soluble. And here are some examples of globular proteins in enzymes, hormones, and antibodies. And then there is fibrous protein, which are long and narrow shapes. So fibrous sounds like a fiber, so it's kind of long, insoluble in water, and they make up used for different functions. For example, they make up extracellular matrix, so they're found in cartilage, ligaments, tendons, and keratins are found in hair. Okay. So some of these functions also include things like, so we just talked about it, but looking specifically, enzymes will be catalyzing reactions. For example, amylase helps in catalyzing the reaction of starch, like or initiating the reaction or conversion of starch into maltose, first step of digestion. They serve as hormones such as insulin that can help reduce blood sugar. They serve as antibodies, which means that immunoglobulins are primarily proteins that help in defense against antigens. And we talked about hemoglobin in a little more detail, which was just for transportation of oxygen and, you know, elimination of carbon dioxide. So what are the other functions? There are other functions such as they can provide structure for the skin. They can help in muscle contraction, and they can help in clotting of the blood. So a lot of functions are shown here, which you can read the slide. I don't have to spend a whole lot of time here. You can clearly see that there is role for signaling proteins in the blood, enzymes, cell membrane, what is the role, structural proteins, what do they actually do, and then proteins in muscles and immune system. We talked about it briefly. So anyone getting their hair done at home or outside, it's a really interesting topic because this is very much related to proteins. You know, there is when you are you know, providing them you can see that your water kind of helps break the disulfide bond, which leads to the, which leads to, just the single sulfurs being present, the sulfur cysteine being present. But then when you are, processing them, and then what happens is that these, let us say you are giving them a certain curling agent, and then you're providing the heat, then the disulfide bond take place, and they are resulting in those perms or the setting of the hair. And you can add a reducing agent, and you can break them by separating those. You can see that these hair molecules, they're just kind of broken now. So there's like lesser curls, or you can add an oxidizing agent, and then you can make those curls again. So I think it's really an application that you might be seeing, or you might have at least heard of, people getting curls or perms, and that is relevant to this and how the cysteine particularly interacts in this. So what is an enzyme? That is the next part and the last part of this, and that's these are the molecules that can catalyze chemical reactions, and they are not sometimes sometimes they work alone, but a lot of times they will need a cofactor, and it could be a vitamin, it could be a metal cofactor, it could be something like that. So why do we actually need enzyme, and how are they different from catalysts? So catalysts are just, it's in the non-living system. These are, enzymes are the ones that are present in the living systems. And if you see here, this is a metal catalyst. That's not really made up of a protein, I guess. And that's essentially what this is. This is made up of a protein, mostly, sometimes. So we're not going to talk about variations, just protein. So these are, again, made up of amino acids, very obviously, and they help in starting a reaction, okay? So what happens is, to start any reaction, you need energy of activation. And what enzymes do is they will reduce that and will enable the reaction to start. So just in other words, if we want to say, catalysts do convert reactant to products. And when we are talking about enzymes, instead of saying reactants, we would just say substrate to products. And so what happens is, when you have enzyme and there is a substrate, then it combines. Sometimes there is this intermediary complex, which is the substrate enzyme complex, and then it leads to the formation of the product. So this is a very generalized view. I mean, there is a lot more that goes, but we're just trying to see an overall picture here, right? So there's an enzyme substrate complex that is formed, which will result in product. But the most important thing also is that the enzyme is released. It is not really consumed in the process. It doesn't really, it gets utilized. It does the process, and then the substrate is converted to product. And here are some nice examples in here, that enzymes that can break down or do different things and convert, and most of them feel like break down in here. So that's what it is. So what are the properties of enzyme? Why do we actually need the enzyme? So you will see here some really good examples. So first of all, they can make the reaction go much, much, much faster, like 10 to the power 3 to like 1,000 to more than 1 billion fold faster. And they're highly specific, so it's not like they can make mistakes, because it's based on a lock and key kind of mechanism. And they have a certain set of conditions, like temperature, pH, and all that, under which they work. So what happens, why did we say it makes a reaction faster? The reason we said that was because without enzyme, the substrate may find it very, very hard to get into product. But what the enzyme does is, it really helps in getting them together. It reduces that energy. So now it is much easier to like kind of take this ball all the way instead of up the hill, very little bit, and then that can result in the desired products. So how do the enzymes work? By lowering the activation energy. That is the most important thing when you talk about enzymes. The function of enzyme is to lower that activation energy. So how fast the enzyme work? A lot fast. Actually, human body will not even work if you didn't have certain enzymes. So for example, chemical reactions that are essential in creating the building blocks of DNA. There is a certain reaction that would take 78 million years without enzyme. And it takes 10 milliseconds with the enzyme. So enormous change in the time, like, who has that kind of time to wait on? So the enzyme catalyzes the reaction, it speeds up the reaction, and it helps in establishing the product very quickly. Another example is carbonic anhydrase that catalyzes the hydration of carbon dioxide to speed up its transfer in like water-like environment or like blood. So it's one of the fastest known enzyme. And if the enzyme is not present, one molecule will be, created in, the hydration will happen in 100 seconds. But with the enzyme, there is 10 million molecules are processed in 100 seconds. So each molecule of enzyme can hydrate 100,000 molecules per second. It's 10 million times faster than the non-catalyzed reaction. So just stressing the importance, like our bodies could not have done without the enzymes, would have completely failed, because the processes that we are doing, the rate at which we are doing is very fast. And that cannot be sustained by non-enzymatic reactions. For the last part of classification, we have 6 different groups. One is Oxidoreductases, which catalyze the oxidation process or the reduction process. So some of the examples are the lipid oxidation, adding an oxygen to the fatty acid, or oxidizing phenols in the food. Those are some of the oxidation important reactions. Now, why we put them together? Because these happen together. When something is getting oxidized, other thing gets reduced. So they are happening simultaneously. Then there are transferases. As the name suggests, it is responsible for transferring a particular part, let us say a certain group, from one substrate to the product. Okay, so we are going to see a pictorial representation of that. So if you see here transferases, you can see here this yellow part is transferred from here to here. And so this is the example of transferase. And like I said, oxidoreductases, one thing is getting reduced. One of them is getting oxidized, so it gives away. And the other, which accepts these electrons or the hydrogen or whatever the context is, it gets actually reduced. So you can see how redox reactions are always happening parallelly. And you saw the transferases too. So now let us take a look at hydrolysis. Hydrolysis is, like we already talked about it in module 2, 3, not really in 4, I think, is the breaking things with water. And some of these reactions are really important because they are helping in carrying out different steps. So example, there is amylases, which cleave the glycosidic bond, which we talked about. There are proteases, there is lipases, all of these are breaking of the bonds. Then there are lyases, which is not to be confused with transferases. They catalyze removal or addition of groups. Okay? So you are adding certain groups, or you're removing certain groups. Okay, we're going to see an example in this one. So if you see here, there is there is a substrate. These are the substrates, and they can be combined together, because this is what you are actually adding to this B. And so there are different examples here. Like for example, there is carbon-carbon or CO or CN, or different kinds of lyases or synthases that are possible. That's another name of that is synthase. Isomerase. So we talked about isomers. So you can imagine that isomerization would be rearrangement in different ways. So you can think of it, you can think of RLND isomers, and that is one possible isomerization reaction. And it's also possible in more than one context. So you can see that there is it just is rearrangement. You can see how this was in the middle, and this was here, and this is now rearranged, and essentially it kind of looks the same, but the structure is not really actually the same. And finally, the ligases, which is its ligation is combining. So it is combining of substrates with the help of ATP. So you can see here A and B, and with the help of the enzyme and ATP, it kind of gets combined to make it one. So these are all the different reactions or enzyme classification. And these enzymes, they are very prone to be affected by temperature, pH. What is the substrate? By substrate, I mean, again, the initial, molecule that you're starting with. What is the concentration of enzyme? What are the coenzymes and factors? So these are all the things. So the use of enzymes in clinical medicine, I'm not too worried about this. I think that this is going to be something that you can kind of read through. So they're used in diagnosis and prognosis of diseases. They're used as in therapeutic purposes, and as of course in diagnostic. So here is an example that is given. This is just for you to get an idea. For example, plasma has a lot of enzymes, and if you are looking at, let us say, the doctor asks you to get a blood test done, and they're looking for certain enzymes, or they're looking for a level of certain substrates, they can tell, oh, there is too much of this in blood. That means this enzyme may not be functioning, or may not be working, or maybe overworking. So plasma enzymes, there are two types. One is functional enzymes that are synthesized in the liver, and they're present in the blood in high concentration. And then there is non-functional enzymes. These are just present in low levels, and they don't have a function, but if they are elevated, if there is something happening, then you would just see a lot of non-functional enzyme, which would indicate that there is something wrong in a certain organ, or some certain disease state has occurred. So here are some list of enzymes that are used for diagnosing different things. If you would see, if you're looking for certain diseases, you would be in turn looking for upregulation or downregulation of certain enzymatic activity, which means that if you think there's increased activity of amylase or lipase, it could be an indication of pancreatitis, and similarly you can see for the other ones. So they're used in diagnosis because their isomers can be used in diagnosis because they're different forms. And sometimes the indication of one form may be indication of a certain disease state happening. And so there is isoenzymes of their clinical applications include LDH and creatine kinase. You'll see in the next page and the next slide. You can see here that there are certain isomers, LDH1, LDH2, these are all isomers of lactate dehydrogenase. They're present in different tissues. And if there is a LDH increase, that may be indication of a certain disease state. And that's, you can look at certain LDH increase, and that would indicate specific things that you need to know about a certain disease state. So when you come across studying of more of the, I guess like a pathology kind of course, or where they're looking at certain enzymes, some of these areas may be highlighted, some of these enzymes may be highlighted depending on what exactly you're studying. A similar example is creatine kinase. It's a dimer, it has two subunits, and there is, they have different presence in brain or heart or muscles. And if you see in a certain infectious state, myocardial infarction, you can see that both CK and LDH are increased in level. And then they can also, I mean, you can see the differences in level, and after treatment, like what happens, and or at different stages of diseases, what happens, that is an indication of how well the body is working. They're also used in therapeutic agents. I think we shared briefly that they can help in, their blood clotting is one part where they're implied in. So they can help in clearing blood clot. They can help in controlling levels of certain enzymes or certain substrates, and that's where the therapeutic uses are. And similarly in diagnosis, you know that there is they can determine the glucose or uric acid or urea or cholesterol, all of these they can be used for. So a lot of application in health. I don't really see a whole lot of application directly in dental, thing, but of course there is an overall health factor where there is a lot more application or in nursing, a lot more application if you are doing that. Okay, so we have the only thing that I found was like, okay, so there is, just like we talked about lipids. So in saliva, there is enzymes, of course, and there are certain proteins which are more importantly present. And you can see some of the names that are indicated here. So that's about it for, it's right around 3:30. Let me stop sharing. And also start.